Yes, am I audible?
Yes, sir.
Yes, sir.
Okay. Okay, good morning, and let's continue, and we will have a quick review of what we have learned last time. And our focus in the last class was to understand the experiment they performed, or their observations from which the discovery of restriction and modification systems has happened. And that is about EOP, and EOP is for efficiency of plating. We discussed about plating, and we discussed about what is K. I mean, there are two strains, E. coli K and C. We know what is lambda K. Lambda phages that have grown on E. coli K strain are referred to as lambda K. Similarly, lambda C, which are grown on lambda in E. coli C strains. So E. coli K are good. That means, in the sense, the lambda K strains they always have efficiency of plating of 1. They're fully potential. Whereas lambda C is also potent. The lambda C is also potent in with EOP of 1 when infecting E. coli C. But infecting E. coli K was deficient, and there is something about it. It is because E. coli K strain has a restriction and modification system in it. So let's see it as a continuation from what you have understood in the last class. And E. coli K strain has R&M system, one of it, whereas E. coli C does not have. Now, if a lambda was grown on it, it must be having some sites within its genome. And those genomes, if they are already grown, that must have been methylated by because they are produced in lambda K strain, the methylase would have added methyl group. So they can infect E. coli K strain very well, because they are now resistant to the restriction endonuclease, because the site-specific sites are protected from restriction. E. coli C does not have any restriction endonuclease, so basically any kind of lambda would have been able to infect it successfully. But now, if the lambda K was grown on E. coli C, they would become lambda C, right? We call them as lambda C. And now the restriction sites they have within them, their genome, remain unprotected, because E. coli C does not have any R minus M minus. So they can infect it, infect E. coli C, but they cannot be methylated, and also because there is no restriction, and the lambda C that is grown in E. coli C will not have any methylation compared to this one, lambda K. So now when lambda C, we try to infect lambda C onto E. coli K, K has restriction site, restriction enzyme, gene for restriction enzyme, and the methylase. But then, because the restriction endonuclease is present, the genome upon injection into E. coli CK would get cleaved off. And the argument can also be that there is methylase, and why should it be only restriction endonuclease? Usually there could be multiple restriction sites, right? Multiple sequences which this R restriction endonuclease or methylase could be recognizing. So there is a probability of R or M recognizing. The same is the case with all. This is just for thinking purposes, and so. Methylation should have been done in all the places. Only then this genome will be able to exercise or complete the infection here. Even if in one place there is a cut by the restriction endonuclease, then it cannot. It is made, it has become into 2 pieces. So upon replication or so, you will not get complete lambda phage. You will not get. So even if you think of probabilities, assume that methylase has, there is equal probability, assume that each of these enzymes could be acting on each of these restriction sites, either methylation or restriction. Even if in one place there is a restriction or endonuclease acts upon and breaks it, that phage cannot infect any further. Okay, that is how these R&M systems are very effective. So EOP should have been 1, but it is 1 in 10,000 only can be. That means maybe in one of those, that out of 10,000 may have a probability. That's a very less probability that all the 4 restriction sites, for example, must have been methylated, or somehow the restriction endonuclease may not have been accessed, may not have accessed these, and some kind of escape may have happened, and that is what 1 in 10,000 phages may have infected the E. coli strain, and only then you can get lambda K. Okay, this is just a little bit extension from what we have learned in the last class. So we also discussed that it is not possible to have R&M system because the genome could become vulnerable if there is no methylase gene, and the restriction endonuclease will break it, and the genome would be useless and the cell would die. We discussed about type 1 and different types, type 1, type 2, and type 3, type 2S as well, out of which type 2 is the best one because R and M are on separate subunits, separate enzymes. They do not require additional cofactors. This recognition sequence is defined and symmetrical, and they cut within the site. That will add a lot of control to what we are doing. The certainty is established, and some of them could produce staggered ends, which could happen, which could be sticky ends, which we will discuss today. I think that is where we left, I guess, in the last class. So yeah, we also discussed about the nomenclature, and I don't think there is requirement for any more italics, but the rest of the things will remain same. That is, we will have the capital letter for the genus name, the 2 alphabets from the species, then the order of discovery. MA comes from the genus. Species. S comes from genus. MA comes from species, marsensis, and this is the Roman number 1. That's the first enzyme discovered in that particular organism. So if you find any sequences like this, GT for HIN2, for example, they put PY, PU, AC. So PY is pyrimidines and PU is purines. A or G, any of them could be there, and here T or C. Okay. And in some cases, you may have CA, NNG, CC. For example, I'm just making GGNNCC. N would represent any of the bases, so any of them could have been there. In some cases, you will get that also. So the enzyme recognizes these specifically. And in this case, the enzyme recognizes these 2 specifically, but may require spatial constraints for purines and pyrimidines as indicated here. We also discussed about 6 base cutters, 4 base cutters, and 8 base cutters. We will discuss further a little more in today's class. And all the recognition sequences are in palindromes. That is, whether we read from here to in this direction, always reading should be from 5' to 3' in all of molecular biology, and 5' to 3'. So in this slide, I would also want you to remember GAA-TTC. Let me write it again. 1 second. Let me try. GAA-TTC. GATTC is not equal to CTTAAG. Okay, don't keep it this way. If you're saying this would have been 5' to 3', and this would have been 5' to 3'. So these two may look palindromic in a way, but they are actually different. Unless if you had a sequence, a complementary sequence like this, GAA, TTC, and it is mentioned that this is from 5' to 3'. Normally, nobody writes 5' to 3' in this direction, unless it is a complementary strand of something, or there is a, as you will see in some of our things that we will discuss, there may be some difficulty for the diagram, putting it as a describing how things happen that time. But otherwise, if no mention is made, GATTC, it is implied that the sequence is read from 5' to 3'. Okay. And here is a nice diagram about how restriction endonucleases interact with DNA. You'll also see another picture, and then they make a cut as two subunits. They bind as dimers, homodimers, and then they cut like this. If you see it from a different dimension, this is BAMH1 endonuclease, and this is the DNA as a helix is here. Inside, you can see that there are the nitrogenous bases. Outside, you have the phosphate backbone. And the BAMH1 endonuclease homodimer is sitting on the DNA, and it will cut the DNA at the locations it should. I also asked you to remember two for EcoR1 and BAMH1, right? So that whenever we are using, you remember this GAA, and BAMH1 is GGA. I'm saying only 3 bases because the rest can be complementary of this TT, and for G I put C. Here T, C, C. GGA TTC for EcoR1, and GAA TTC for EcoR1, GGA TCC for BAMH1. Try to remember those two. We also discussed about the axis of symmetry. We also discussed about what is meant by this asterisk and what is meant by this slash. This is the site of cut or restriction. That is where the endonuclease activity would have happened. Cut would be made here like this, right? This will go as one part, and the other ones will go as another part. I discussed about the asterisk. That is where the methylase or modification enzyme would work, and it's usually methylation. And in this case, at least, methylation happens on the amino group of adenine. So types of restriction enzymes, I think that is where we must have started. We also have discussed about what is meant by an overhang, and we will see a little bit of it today. We already saw an GAA TTC, and I want you to also have a book and pen in your hands because there are 2 or 3 small problems, very small ones, and I want you to solve them today. So please have a book and a pen with you, or a pencil preferably. CTTAAG. And this is a DNA molecule. And if you remember, the slash is put somewhere here, typically. Okay, let me try this. So slash is somewhere here, right? That is the site of cut. So the cut, when it makes, these are the hydrogen bonds that would be broken. This DNA will go separate, and the other DNA molecules will go separate, something like this. If they are going and you separate them, you will get G, C, T, T, A, A. That's one DNA molecule. A, T, T, C, and G. And always the top strand, typically the top strand is 5' to 3', and the lower strand, when you are representing in textbooks or when we are writing as answers, that's how it's usually done, unless there is a requirement. But otherwise, it's if nothing is mentioned, the top strand is 5' to 3' typically. So this would be the 3'. This is 5'. This is 3', and here you have 5'. This part, TTAA, is the overhang. And it is the 5', because the 5' side of it is having the overhang. We would have referred to it as 5' overhang. Now, I would also want to look at PAC1. There are many other examples that will come. TTAA, TTAA. This one has a different architecture, or let's see. AATT, AA, and TT. And it cuts somewhere here. So if you cut it and separate the two DNA molecules, you would have TT, AAT, and AAT. And here it would be TA, ATT, and TAA. Right? So 5', 3', 3', and 5'. So in all of the any of the question papers, including CIAs, assignments, and even in final exams, please don't forget to put the primes, 5' to 3'. And I'm sure some of you are used to call this as dash, right? I think some people used to call it as 3' dash or 5 dash or 3 dash. I think dash is not a correct word to use that way use. I don't know if it is from maths or somewhere. I am unsure about it, but you call it as 5' and 3'. From textbook, I'm saying. Okay, and I hope you know what is meant by 3' and 5'. The chemistry of it, please try to have a look at it all the time. That's important. So we will say, I will quickly draw deoxyribose, maybe 1, 2, 3, 4, and you have CH2 OH. So if this would be ribose, right? Roughly the structure of ribose. And you would have had a base here, adenine or so. 6-aminopurine is adenine. So let me try to put it. I'm roughly trying, and you have NH2 here. So this is 1, 2, 3, 4, 5, 6, 7, 8, and 9. And when I said about methylation, this is where the methyl group would have been added by methylase. So there are double bonds and other things, you adjust those. I'm not drawing that. So the numbering of nitrogen bases is given as 1, 2, 3, 4, 5, 6, 7, 8. And now you have to give for ribose or deoxyribose. Say we will call it as deoxyribose. I removed this is 1, 2, 3, 4, and this is the 5th carbon. How do I distinguish between the 3 position of nitrogenous bases and 3rd position of ribose or deoxyribose? We need to have a difference. Therefore, they added this 1', 2', 3', 4', and 5'. Okay, so, and every time we say about 3' OH, this is the meaning of it. And if you're saying 5', it's actually a phosphate group that would have been here. right? Phosphate with all the other stuff, and it would have been, this would be forming the phosphate backbone, phosphodiester bonds. And the inside of the DNA would be these. So when we say 5', it means the 5' phosphate group. And when we say 3', it means the 3' OH group. Both are important for DNA and nucleic acids, basically. So I hope it's worth revisiting. If you're missing any of the fundamentals, it's worth revisiting it to the very basic things. Okay. Otherwise, I just put 5', and if we don't know what is that there in that, then it will be a little bit trouble. So 5' phosphate group is there, and 3' OH group is there. Okay, so I hope the point I'm getting to here is this would be 3', this would be 3', here is 5', and this one is 5'. You see a overhang of AT, and here also you see a overhang of AT, and that is the 3' overhang. And if you see any of the other ones, say for example, you're going to see SMA1, and I will discuss two of them and then we'll move ahead. For SMA1, you see CCC and GGG. It is plain and simple. GGG, CCC, and the cut is here. And when they separate, you will get GGG, and here is GGG, CCC. So there are no overhangs, and these are referred to as blunt ends or flush ends. Flush is something like clear. And I want to take up one more, that is SAU3A, because it shows it dash there. It actually means that there is DNA, something like this, GATC, and you have GATC. That's the DNA molecule. Cut would be made here and here. So upon cut, one would have C, T, A, G, and you will have, GATC. The DNA would have been something like this, and it would have resulted in 5' overhangs. This is 5', but this is 5', this is 5, and so on. Okay, I hope that is a good enough explanation for those who have not studied genetic engineering before. So this is the same thing represented here, sticky ends. When cut is made, you will have two separate DNA molecules like this. That is 5' sticky ends or overhangs. Why do you call them sticky? Because they can form hydrogen bonds. They are still complementary, and they can form hydrogen bonds. And blunt ends or flush ends are those that have clear without any overhangs. So it's simple, right? We'll go through, try to identify the types of ends they would make. This would be, or tell me all those that have 5' overhangs. That is, if this is the symmetry, axis of symmetry, if the cut is here, then it is likely to generate a 5' overhang. If the cut is on this side, then it is generating overhang. If the cut is exactly on the axis of symmetry, then it is going to give blunt ends. So one, MBO1, MSP1, and BGL2, CLA1, and NOT1. They are all going to give 5' overhangs. Blunt ends would be ALU1, HAE3, and PVU2, and so on. And 3' overhangs will be the other ones. You don't need to by heart all this stuff. Okay, I'm just saying, every time, if I'm giving a question, then majority of the times I give the required information for you. Your memory is not what we want to test. We want to test your ability to troubleshoot or come up with a design such that the required condition is met. Okay, how good you come how much you can use your cognitive skills is the objective of this course, not memory. Not that you shouldn't know. I mean, everything is a memory. If you have learned about an idea, it is intelligent idea. But once you remember it, it is memory, isn't it? So everything becomes memory eventually. But I don't need you to remember all these stuff. Anyway. So there are some things about, we need to discuss some technical terms like called as isocytomers, neocytomers, and so on. Isocardomers also. Isocytomers have the same recognition sequence. I mean, sequence specificity is same, and also the cut site is same. If there are two different, say for example, SPH1 and BBU1, they recognize same sequence, CGTACG, and the same thing here also, CGTAC, and their cut site is also same. So they are referred to as isocytomers. Another example is ACC 651 and KPN1. The recognition site is GGT-ACC. Same is the case here to KPN1, GGT-ACC. And their cut site is also, no. We'll go, their cut site is different, right? I'm sorry about it. For isocytomers, at least, this is the answer. Okay? For neocytomers, I have to check this with the textbook. So if you have learned from Primrose, there could be differences from internet. There are several other differences as well. The textbook, prescribed textbook here is Genes to Genomes. I think I've picked that because it's very easy to read. So with this, within this methodology, there has been differences, but to my understanding or memory, this is what I always follow. Same sequence specificity and same cut site is isocytomers. Neocytomers are same sequence but cut at different points. Something the examples that are given here actually represent the neocytomers. We will get back to it once we will look at the internet again and see what is the actual definition. There has been discrepancy between textbooks. The examples that are given here for neocytomers, where you have the recognition sequence is same in this case, but the cut site is different. Okay, in one case the cut is here, in the other ways there is a difference, right? One is generating a 5' overhang, whereas the other one is generating a 3' overhang. The cut sites are different. Here also we have SMA1 and XMA1, and the recognition sequence is same. SMA1, as we saw earlier, will cut in the axis of symmetry, and it will generate blunt ends, whereas with XMA1, the cut site is here, and it will generate 5' overhangs. Okay, let me make this clear. Isocytomers, as I said, we will have a look at it or verify on the internet. These are all just technical terms. We don't need to really mug up, but it's worth knowing that there could be different enzymes in different organisms that recognize the same sequence and cut at the same sequence. And there could be different enzymes that recognize the same sequence but cut at different places. That's the essence of what we should actually learn. And so ISO is for same. Same sequence, same cut site is isocytomers. Neo is new. They recognize the same sequence but cut at different points. Okay, something like that. We'll have a relook at it later time. Methylation dependency is another point. We discussed about R and M systems, restriction and modification systems. And you will also find there are diverse restriction endonucleases, R and M systems, and some of them have an issue. Say HPA2 and MSP1. They both recognize the same sequence and cut at the same site. But there is a problem. Is some of them, like for example, the MSP1 can recognize both methylated and unmethylated one, or can act upon, say for example, you have, this is the sequence, and if the second C in this one, if it is methylated, MSP1 can recognize and cleave as well, whereas HPA2 cannot cleave methylated DNA. So I'll repeat again. MSP can recognize methylated and unmethylated DNA, this sequence, and cleave it. Whereas HPA2 cannot cleave if it is methylated, but can cleave if it is unmethylated. Okay, so I actually call them here, I actually call them, there are three different ways I describe them. Let me try to correct it. So methylation sensitive, I call it as methylation sensitive, methylation insensitive, and methylation dependent. This is why I'm taking time to describe it, is because sorry, we will discuss about this as a part of epigenetics or DNA methylation. One second. Sorry. Sensitive here is if the sequence, target sequence is methylated, the enzyme cannot work. Okay. Say out of this HPA2 will fall into this category. Insensitive means irrespective of if that sequence is methylated or not, it can cleave. And that is, in this case, you will have MSP1 as the insensitive one. It can cleave whether it is methylated or unmethylated. Whereas dependent is one enzyme is called there, DPN1, that will cleave only if its target sequence is methylated. It cannot cleave hemimethylate. It cannot cleave if the sequence is unmethylated. So when I say this, there is also one more point I have to discuss, is if what will happen if the first cytosine is methylated. In that case, maybe MSP1 also cannot work. Okay, I want you to know these things a little bit clearly. That is, the methylation profile of the recognition sequence can influence the activity of the restriction endonucleases. Just keep that in mind. In that category, it is dependent on whether they are sensitive or insensitive to that particular position being methylated. Okay, and some enzymes like DPN1 will cleave only if they are if the target sequence is methylated, else not. It recognizes a different sequence. I think it is GATC. We'll get to those finer details again tomorrow when we do the actual problem as well. I will repeat these things at that time. So for now, if you understood that there are enzymes from different organisms which might be cleaving the same site, same recognizing and cleaving at the same site. There are enzymes which might be recognizing the same sequence but cut at different sites. And there are also different enzymes that will differ their activity based on the methylation status of the target sequence. Okay, these are all going to be useful, and that's why we should know them. Number and size of fragment, I have, yes, number and size of fragments. This is also very fundamental. We all must have known this, but we will just go through it for the sake of people, others. I'm just putting any base. What is the probability of this? In each position, there are any of the bases could have been there. How many bases are there? 4. In this position, 4. And you have 4, and here also 4. That will give you, if you multiply 4 into 4 to the power 4, then you would probably get the diversity or the possible combinations. Okay, all the possible combinations. And GATC, for example, would be 1 in how many bases would this be? Somebody can calculate and tell me.
Like the 4 power 4 combinations. That's what you're asking, sir.
Yes.
It's 256.
Okay, it is. Out of all these 256 possible combinations are there, and out of 256, GATC, if you are looking at it, is 1. That means there is a probability of 1 possibility of GATC in about every 256 possibilities if you are looking at this 4 base combination. If you want it at 6, if there is 6 bases, 6 base combination, then you will have something like this. That would be into 4 into 4. That will be a probability of 1 in 4096 possibilities, right? So for every 4,996 bases, you might have 1 possibility or probability of occurrence of a one particular type of sequence, say for example GGATCC, right? I hope you understood what you understand, what I want to say. So let me try once again. So there are some things you have to remember. Say for example, there is a 5 kb DNA and it has 100% GC content. Okay, GC content. If I said 100% GC content, that means 80 is 0. Then is there a possibility or probability of this site occurring? No, because 80 is there. I'll also put it this way. So if I say there is 50% GC, 50% GC content, then the rest of it is 50% 80. Then there is a possibility of occurrence of a GGA TCC, BAMH1 site, right? Because they have GC as well as AT. But again, that doesn't help us, because if I said all the G's are on this side, GC is here, and all the A and T's are here, then is there a probability of GATC occurrence? It is not there. So in problems that we actually discuss, it's worth remembering these, that first thing we should probably have 50% GC content. It's not that it should be like that all the time. Say for example, if you look at this site alone in BAMH1, it's say you have more of GC than AT. It's just 1/3 of it, isn't it? So that percentage could vary. Don't worry about anything that I say. See if you can understand some numbers and view them. The second thing, first point you should remember is that there is equal proportions of all the bases represented to ensure that all are represented. Second thing is we should see that the bases are distributed randomly along the DNA. Then say for example here, if you have, then probably you will have all the things represented randomly. Then the numbers that we are talking here, say 1 in 256 if it is a 4 base cutter, 1 in 4096 if it is a 6 base cutter, right? Most often we use in restriction endonucleases, we use the 6 base cutters because they give sufficient or big enough size of the fragment. If we are using a 4 base cutter, we will use them as actually at purposes where we need, but otherwise the fragment size is too small for a gene to be cloned and so on. right? Because we will get to it. Say if I want to clone a gene, I took the gene and I had something like SAU3A, and here also. If you say this gene is about 2 kb, then the probability of occurrence of SAU3A within this coding gene is very high. And if I use SAU3A to cut this, there is a likelihood that SAU3A might also cut the gene in between, right? So to decrease the probability of occurrence, but at the same time, have it not make it too rare, then in that sense, if you see 6 base cutters are actually more useful to us. Okay. One second. So to see if you take the genomes of different organisms and you cut the DNA, I hope this table now will give you a better understanding of or complete understanding of what I'm trying to imply also. Say for example, yes, if we have APA1, which cuts mostly GG and CCC, the Arabidopsis, the number of sequences that it would give, sequence fragment size is 25,000, average of it. Typically, what should it be? It should be about 4,096 if it is a 6 base cutter, right? All these are 6 base cutters. But then the sizes they give, the fragment size, average fragment size of these genomes from the plant Arabidopsis thaliana, nematode, Drosophila, E. coli, and humans. So these are the ways, because the bases are not randomly distributed. There is sometimes you may have high GC content, sometimes you may have less GC content in some places, and so on. So this gives the understanding for 2 things. One is the GC content, and the second one is the probability of occurrence, and also the fragment size. Yeah, and also the base composition within the target site itself. If you have only GC, then you're having less in some cases. In plants, if you see it's in this way, right? In humans, if you see it is the fragment size is decreasing from decreasing to increasing like this. So I hope it gives a some idea about what actual living organism you should appreciate. Try to understand that the base composition within genomes is not absolutely random. There is some degree of clustering, you can say. So one time where you will see some places very rich in GC and some places very rich in AT. Here is the question I want you to solve very fast. So for this reason itself, we gave in this question that it's 50% GC content and random distribution. They usually mention that way. So assuming a DNA molecule, please take your papers and pencil and try to solve it and tell me the answer. A DNA molecule with a 50% GC content and a random distribution of the 4 bases. A circular DNA of 20, you should also keep in mind, 2048. What is the approximate number of fragments that could form by cleavage of this DNA with a 4 base cutter? Anybody? Two 048 base pairs, and if it is a 4 base cutter, it would usually give about 1 in 256. So from one cut site to another cut site, you may have 256. You will go multiple times, and then how much would it be? 1 cut,
8.
2, 3, 4. Yeah, you can say something like that, I guess. 256.
8, sir.
Yeah, then the answer is 8. But the process is you cut one place and then have 256 bases. These are just theoretical problems, okay? It is seldom that way it happens in real scenarios. Okay, this is simple question. So in a restriction map, you will get an assignment on restriction maps. There will be multiple assignments in this course, okay? All the assignments will make you think. It's only a thinking thing. It's not like you can find the answers somewhere in the internet. You'll have to work it out. So the best source of help is your own classmates. I would encourage those who don't know to seek help. I would encourage those who know to extend their help. I'm not going to discuss restriction mapping much, but I'm going to ask you a lot of it. And at the end, if there is still a need, then probably I will tell you how I do. With restriction mapping, it is about how different people visualize and imagine, right? Different people solve things differently, and it takes time also. It's not like I can take a problem right now and I can solve it. Restriction mapping doesn't work like that for me also. It takes time. It's about we practicing a lot of those restriction maps. I think in the introductory class or in the second class, I told you, please play more games of something like Minesweeper, Sudoku. That is engaging your brain continuously to get something out of it. I mean, that is one essential characteristic or trait every successful people have. If you can't keep your mind engaged, and it's not easy for anybody, of course. And slowly, first thing is to increase your let me put it this way, increase the time of engagement. How long can we keep thinking about it? Second thing is about speed. How fast can we think and achieve? These two can be done using games, video games, something like Minesweeper and Sudoku. Once you get that capacity of thinking for long, and then that is when you will be, it is like being bitten by a bug called logic, and it will make you a different person completely. I'm sure most of you are already, but it's brain and body. When you do this, you're playing physical games like it could be badminton or volleyball, football, cricket, or anything. Brain plus body. If you're just using the brain, what we are doing right now, all of education is just using brain, majority of it. And something like chess or Minesweeper or Sudoku, they will keep you. I mean, chess is a long game, but Sudoku and other things you can play very short. Without moving body, just using the brain, it is not for everybody. All these things, brain as well as body, every animal does. Okay, most animals do. But just sitting there and thinking, that's not everybody's. It's not fun for many people. But that is what we are supposed to do. You're doing mathematics, right? Which I can't do. And if you're learning Maths 1, Maths 2, and all those 4, 5, and so on, that's a lot of engaging yourselves with brain alone. And I want you to take this course in that sense. To solve, to think, but not necessarily in the numbers, but about trying to put together and troubleshoot or get what is required. So it's a challenge between me and you people, where if I give a question, you should be able to answer it very fast and accuracy. Speed and accuracy, that will make your efficiency. Okay, please quickly do this one, restriction map. I will describe what is meant by a restriction map first. Restriction map is, if I am drawing a circle on for here, telling where all the order of restriction sites. There are two things about restriction map: order of restriction sites on a DNA, and second part is the distance between two all consecutive restriction sites. So for example, if I'm going to cut this DNA using restriction enzyme A and B, I should be able to write AA, B comes, again A comes, and then B. That is first one. You should be telling the order of restriction sites on the DNA. Second thing is I should be able to tell the distance between these two, say 2 kb, say 1.5 kb, or 1 kb, 2 kb, and say 3 kb. The data will be given something like this. You should arrive at the answer something like this. So two things should be there: order of restriction sites on the DNA and the distance between every two consecutive restriction sites. That is a restriction map. If you have already solved for this problem that is given here, and you should always be clear about whether a linear DNA is given or a circular DNA is given. Can anybody answer this? Okay, we have only 1 minute. With A, if I cut, I will discuss this again in the next class also. It's giving 3 fragments, so because it's linear, it should be 2. It could be 5, 8 like this. It could be 2, 8, 5, or it could be 8, 2, and 5. Think of all possibilities and then slowly narrow it to what is the correct answer. Don't try to just go with this and then directly go into the next solving to the next ones. Think of all possibilities and then you will arrive at an answer and that will be the correct answer probably. Once you have finished a restriction map, say, you can come up with answers for the next class about this one. Please ensure that all the 3, how to verify whether your answer is correct or not, is once you got a restriction map like this, see if all these conditions given are met or not. Don't just look for 1 or 2 and then say that is the answer. All the 3 conditions should have been met with your final answer. Then it is probably the answer. Okay? Yeah, I'll stop now. We will discuss more on restriction mapping and other enzymes in the next class. Any questions? Any questions? If not, we'll take leave then. Take care and have fun.